Welcome, curious minds, to the deep dive. Today, we're embarking on an extraordinary journey into, well, one of your body's most unsung heroes, the digestive system.
It really is amazing when you think about it.
Totally. Imagine this. You enjoy a meal, and without a second thought, your body expertly absorbs an astonishing 95% of the fats you just consumed. How does it manage such an incredible feat?
Yeah, it's far from simple. It's this really intricate ballet of transformations, all carefully orchestrated and incredibly precise.
Right. And for college and medical students diving into Boron and Boulpaep's Medical Physiology, Chapter 45 on nutrient digestion and absorption, well, it can feel a bit like a dense forest.
Exactly. So
Yeah.
our mission in this deep dive is to be your guides. Demystifying this complex material. We'll break down every concept basically from the ground up.
And try to paint a clear mental picture without needing diagrams, always connecting these fundamental processes to real-world clinical relevance.
That's the goal, making it conversational but accurate.
Absolutely. And you got to think of digestion not just as a stomach thing, right? It's this carefully choreographed sequence that begins even before the first bite, with the sight, smell, taste of food.
It's a highly integrated, finely regulated system, essential for survival. And, well, as you might guess, when any part of this intricate system falters.
The consequences for nutrient assimilation can be profound.
Yeah, definitely.
So let's unpack this crucial chapter, starting with a familiar energy source, carbohydrates.
Okay, carbs. They're a cornerstone of our diet, supplying, about 45% of our daily energy in Western diets? But here's the key physiological challenge. Your small intestine can only absorb carbohydrates in their simplest form.
Has monosaccharides, single sugars.
Exactly. So all the complex carbs like starch, and even the simpler disaccharides like lactose from milk or sucrose, table sugar, they all have to be carefully broken down first.
And while we're on carbs, maybe quickly mention fiber. It's non-digestible for us, but still really important for, digestive health.
Oh, absolutely. Fiber plays a huge role, impacting stool weight, transit time, and maybe even risks for things like colon cancer. It shows that not everything we eat is just about energy.
Good point. Okay, back to the digestible ones, like starch. How does that breakdown happen?
Yeah, it's a multi-step process. It starts minimally in your mouth with salivary amylase, but the real heavy lifting, what we call luminal digestion, happens in the small intestine.
And that's thanks to pancreatic amylase.
Primarily, yes. Pancreatic amylase. Think of it like molecular scissors snipping internal bonds within those big starch molecules.
But it's selective, you said, an endoenzyme.
That's right. It cuts starch into smaller pieces, oligosaccharides, but it leaves the very ends of the chains and these specific McWigo-6 branch points untouched.
Ah, okay. So the products aren't quite absorbable single sugars yet. You get things like maltose, maltotriose, and these alanine dextrins.
Correct. And that brings us to the next crucial step, membrane digestion. This happens right at the surface of your intestinal cells, the brush border.
And that's where specific enzymes live.
Exactly. Three key ones: lactase, glucoamylase, which is often just called maltase, and the really interesting sucrase isomaltase complex.
Okay, wait, sucrase isomaltase is two enzymes in one.
Pretty much. Lactase, well, that breaks down lactose into glucose and galactose. Yeah. Sucrase handles sucrose, splitting it into glucose and fructose. But the really critical part is the isomaltase.
Why is that one so critical?
Because it's the only enzyme we have that can break those stubborn I-16 branching linkages found in the oligodextrins. Without isomaltase, starch digestion wouldn't be complete.
Gotcha. So once these brush border enzymes do their job, we finally have our simple sugars, glucose, galactose, and fructose, ready for absorption.
Ready for absorption. And how do they get from the gut lumen into the bloodstream? It's basically a two-step dance across the intestinal cell, the enterocyte. First, across the apical membrane into the cell.
And then out the other side, the basolateral membrane, into the interstitial space and towards the blood.
Precisely.
So how do glucose and galactose get in on that first step?
They primarily use a transporter called SGLT1. This is a fantastic example of secondary active transport.
Okay, what does that mean exactly?
It means the sugar molecule can only get in if a sodium ion comes along for the ride. It's like a co-transporter. The energy for this doesn't come from breaking ATP directly at SGLT1, but from the sodium gradient.
Ah, the sodium gradient maintained by the NACA pump on the other side of the cell.
Exactly. That pump keeps intracellular sodium low, creating the driving force for SGLT1. And SGLT1 is quite specific structurally.
Interesting. What about fructose? Does it use SGLT1 too?
Nope. Fructose gets its own transporter, GLUT5, also on the apical side. But this one works by facilitated diffusion.
Meaning no sodium needed, just moves down its concentration gradient.
Correct. It's Na plus INA independent, and it's found mostly in the jejunum.
So different ways in for the different sugars, but how do they all get out of the cell?
They share a common exit pathway on the basolateral side, GLUT2. This has also facilitated diffusion. So glucose, galactose, and fructose all use GLUT2 to leave the enterocyte and enter the interstitial fluid.
A very neat, coordinated system, but I guess it doesn't always work perfectly.
No, and that leads to clinical issues. A very common one is lactase deficiency, or lactose intolerance. Many people, especially from certain ethnic backgrounds, naturally lose lactase activity after weaning.
Right, leading to symptoms like cramps, bloating, diarrhea, because the undigested lactose goes to the colon.
Exactly. Colonic bacteria fermented, producing gas, including hydrogen, which is the basis for the hydrogen breath test diagnostic.
Yeah.
But importantly, these individuals absorb glucose just fine. It shows it's a specific enzyme issue.
And then there's that rarer condition, glucose-galactose malabsorption.
Yes, a hereditary disorder caused by a defect in the SGLT1 transporter itself. These patients get severe diarrhea if they ingest glucose or galactose.
But not fructose, because fructose uses GLUT5.
Precisely. It beautifully illustrates the distinct roles and importance of SGLT1.
Okay, fascinating stuff on carbs. Let's pivot now to the body's building blocks, proteins.
Right, proteins. Our bodies are incredibly efficient here too, absorbing most dietary protein. Very little nitrogen, less than 4%, usually ends up in the stool.
And it's not just dietary protein we absorb, right? There's also endogenous protein.
A significant amount, actually. Think digestive enzymes secreted into the gut shed intestinal cells. These get broken down and their amino acids recycled. It's quite efficient.
So how does protein digestion kick off? Stomach first.
Yep. In the stomach, we have pepsin. It's secreted as an inactive proenzyme, pepsinogen, and gets activated by the stomach's low pH. Pepsin is an endopeptidase. It cuts internal bonds in proteins.
How much does it actually digest?
It handles maybe 10–15% of protein breakdown. It helps, but it's actually not absolutely essential. People who've had their stomachs removed can still digest protein pretty well, thanks to the next step.
Which happens in the small intestine with pancreatic enzymes.
Exactly. The pancreatic proteases are the major players. There are 5 main ones, and like pepsin, they're all secreted as inactive proenzymes, trypsinogen, chymotrypsinogen, prolactase, and 2 procarboxypeptidases. This prevents the pancreas from digesting itself.
Smart. How do they get activated?
The key is trypsinogen. It gets activated to trypsin by an enzyme called enterokinase, which is located on the brush border of the jejunal cells once you have some active trypsin.
It activates more trypsinogen and the other 4 proenzymes too, like a cascade.
Precisely. A very effective activation cascade. Now, these pancreatic enzymes include both endopeptidases like trypsin and chymotrypsin, which cut internal bonds, and exopeptidases, the carboxypeptidases, which snip off amino acids from the ends.
So this pancreatic attack turns proteins mostly into smaller peptides.
Yeah, about 70% oligopeptides and maybe 30% free amino acids. But we're not done yet.
Still more breakdown needed.
Right. We move to brush border and cytosolic digestion. The brush border itself is packed with various peptidases that chop down those oligopeptides further.
Into even smaller peptides. Or free amino acids.
Both. But here's a really important point. The enterocytes can actually absorb small peptides, specifically dipeptides and tripeptides, intact.
Oh, really? They don't have to be broken down completely first?
Not completely. They get absorbed, and then cytoplasmic peptidases inside the enterocyte finish the job, breaking them down into individual amino acids.
How do those small peptides get absorbed?
Through a transporter called pepti1. This is an H+-driven cotransporter, another example of secondary active transport. It actively brings in di, tri, and maybe even some tetrapeptides.
And you mentioned a kinetic advantage.
Yes. Because pepti1 can bring in multiple amino acids bundled together in a peptide with each transport cycle, it can sometimes lead to faster overall amino acid uptake compared to absorbing only free amino acids, each via their own transporter. It's quite efficient.
That makes sense. And what about the free amino acids produced in the lumen or at the brush border? How are they absorbed?
They have their own set of transporters on the apical membrane. It's quite complex, with different systems for neutral, acidic, basic amino acids, and so on. The main one for neutral amino acids is system B0, which is Na plus dependent.
Another secondary active transport needing sodium.
Yep. And then once inside, the amino acids exit the enterocyte across the basolateral membrane, mostly via Na plus independent facilitated diffusion transporters heading into the blood.
So defects in these amino acid transporters must cause problems too.
They do. Take heart and nut disease. It's a genetic defect in that apical system B0 for neutral amino acids. Patients can get symptoms resembling pellagra. But here's the interesting part.
Let me guess. The peptide transporter PepT1 can compensate.
Exactly. Because PepT1 is usually normal, the body can still absorb those neutral amino acids as parts of diet and tripeptides, often lessening the severity of the deficiency.
Same idea for cystinuria, which affects cationic amino acid transport and causes kidney stones. PepT1 helps there too.
Generally, yes, the peptide pathway provides a crucial backup. But contrast that with lysine protein intolerance. This is a rare defect in a basolateral transporter.
Ah, the exit pathway.
Right. So in this case, even if the amino acids get into the cell, either as free amino acids or via peptides that are then hydrolyzed, they have trouble getting out into the blood. It really highlights the importance of both apical uptake and basolateral exit.
Wow, okay. Carbohydrates and proteins covered. Now for the really tricky ones. Lipids. Fats.
Well, lipids, yes. The big challenge here is their insolubility in water. Getting that near 95% absorption efficiency is quite an achievement physiologically.
And most dietary fat is triacylglycerols, tags.
Over 90%, typically. These provide essential fatty acids, which we can't make ourselves. But remember, there are also significant endogenous lipids in the gut. Bile acids, cholesterol from bile, shed cell membranes.
Right, which also need to be handled. So first step for fats.
Emulsification. You need to break down large fat globules into tiny droplets. This starts mechanically chewing, stomach churning, intestinal contractions. It massively increases the surface area for enzymes to work on.
And these droplets need stabilizing.
Yes, they get coated with things like bile salts, phospholipids, cholesterol, and even some digestion products themselves, which stops them from merging back together.
Okay, droplets formed. Then digestion. Does it start before the small intestine?
It does. You have lingual lipase from glands in the tongue and gastric lipase from the stomach. These are acid liposes active at low pH. They start snipping off one fatty acid from tags, mainly at the center 3 position.
Is this important?
It's particularly important for neonates digesting milk fat. And interestingly, short and medium-chain fatty acids released here can actually be absorbed directly through the stomach lining to some extent.
But the main fat digestion is still in the small intestine.
Oh, absolutely. Driven by pancreatic enzymes. The star player is pancreatic lipase. It's secreted in an active form, but it needs help.
What kind of help?
It needs collapase, another pancreatic protein that gets activated by trypsin. Collapase essentially anchors the lipase to the surface of the fat droplet, especially when bile salts are present. Pancreatic lipase also needs alkaline pH and calcium.
And what does it do to the tags?
It primarily hydrolyzes the ester bonds at the outer SN1 and SN3 positions. This yields 2 free fatty acids and a very important molecule, SN2 monoacylglycerol, or 2-MAGA.
Why is 2-MAG important?
Because the fatty acid is still attached at the middle position. This structure is key for the next steps. The pancreas also secretes carboxyl ester hydrolase, which handles things like cholesterol esters, and phospholipase A2, PLA2, which breaks down phospholipids.
Okay, so now you have a mix of fatty acids, 2-MAG, cholesterol, lysophospholipids, all still pretty insoluble. How do they get absorbed?
This is where bile salts become absolutely crucial. These digestion products initially form structures like multilamellar vesicles. Bile salts then interact with these, breaking them down further into much smaller structures called mixed micelles.
Micelles, I've heard of those. They're like tiny transport packages.
Exactly. Mixed micelles are composed of bile salts surrounding a core of these lipid digestion products. They are small enough and water-soluble enough to diffuse across the unstirred water layer, this layer of fluid right next to the cell surface that lipids struggle to cross alone.
So the micelles deliver the lipids right to the doorstep of the enterocyte.
Precisely. At the brush border, the lipids leave the micelle and enter the cell. This can happen by diffusion, maybe some collision and incorporation into the membrane, and also via specific protein transporters.
Like for cholesterol.
Yes, cholesterol uptake involves a transporter called NPC1L1, and this is actually the target for the cholesterol-lowering drug ezetimibe. It blocks NPC1L1. Fatty acids also have transporters, like CD36.
Okay, lipids are inside the enterocyte. Now what? You said something fascinating happens.
Yes, the cell basically reverses the digestion process. It takes those absorbed long-chain fatty acids and 2-MAGs and reesterifies them back into triacylglycerols, tags.
Why undo the digestion?
It's about packaging for transport. Long-chain fatty acids first bind to fatty acid-binding protein, FABP, inside the cell, which helps traffic them to the smooth endoplasmic reticulum. There, enzymes rebuild tags, mainly using that 2-mag backbone. Cholesterol is also reesterified.
Okay, so tags are reformed. Then what?
These newly synthesized tags, along with cholesterol esters, phospholipids, and specific proteins called apolipoproteins made in the rough ER, are assembled into large particles called chylomicrons.
Chylomicrons, big, fat protein packages.
Very large ones, too large, in fact, to enter the blood capillaries directly.
So where do they go?
They are secreted by exocytosis from the basolateral membrane into the lymphatic system, specifically into vessels called lacteals. The lymph eventually carries them into the bloodstream.
Wow. A whole separate exit route just for fats.
For long-chain fats, yes. But here's a key clinical distinction. Medium-chain fatty acids, like those found in coconut oil, are different. They don't really need micelles for absorption, they aren't significantly reesterified in the enterocyte, and they don't get packaged into chylomicrons. They pass directly from the enterocyte into the portal blood, bound to albumin.
Which makes them useful for patients who have trouble absorbing long-chain fats.
Exactly. Patients with bile salt deficiencies, lymphatic obstruction, or other causes of fat malabsorption often tolerate medium-chain triglycerides, MCTs, much better.
Okay, that covers the big three macronutrients. What about the vitamins and minerals? Let's start with fat-soluble vitamins A, D, E, K.
Right. Given their nature, their absorption is tightly linked to fat absorption. They get incorporated into those emulsion droplets, then the mixed micelles.
And then absorbed into the enterocyte along with the fats.
Yes, either by diffusion or maybe specific transporters. Once inside, they associate with the forming chylomicrons and are secreted into the lymph. Vitamin A, for instance, often gets esterified before packaging.
So logically, if you have fat malabsorption,
You're at high risk for deficiency of fat-soluble vitamins. Think night blindness with vitamin A deficiency, bone problems with vitamin D, bleeding issues with vitamin K. It all connects.
makes sense. Okay, what about a water-soluble one, like folate, vitamin B9?
Folate absorption is interesting. Dietary folate is often in a polyglutamate form, meaning multiple glutamate molecules are attached.
And that can't be absorbed directly?
No. It has to be broken down to the monoglutamate form, TGLU1, by an enzyme on the brush border called folate conjugase. This step is actually rate limiting for folate absorption.
So getting that single glutamate form is key. Then how does it get into the cell?
It uses an apical transporter, likely an anion exchange or possibly folate OH exchange. Once inside, it gets reduced to its active form, tetrahydrofolate, THF, which is crucial for DNA synthesis.
Which explains why deficiency causes megaloblastic anemia problems with rapidly dividing cells like blood precursors, and why it's so critical during pregnancy for preventing neural tube defects.
Absolutely, a direct link between absorption, metabolism, and major health outcomes.
Now for the really complex one. Vitamin B12, cobalamin.
Ah, yes, the cobalamin cascade. It's a multi-step, highly specific process. Remember, B12 is only made by microorganisms, so we get it from animal products. It's vital for certain enzyme reactions, including one linked to folate metabolism and preventing neurological damage.
Okay, break down the cascade for us. Starts in the stomach.
Yes. First, stomach acid and pepsin release B12 from food proteins. Then B12 binds tightly to a protein called haptocorin, also called R-binder, which is secreted in saliva and gastric juice.
Okay, B12 bound to haptocorin. Then what?
This complex moves into the duodenum. There, pancreatic proteases chew up the haptocorin. This releases the B12.
And then B12 is free to bind to something else.
Yes, it binds to intrinsic factor, IF. HEF is a glycoprotein secreted by the same parietal cells in the stomach that secrete acid. This B12-IF complex is crucial
Why is the complex so important?
because it protects B12 from further digestion, and critically, it's the form that can be recognized by specific receptors way down in the ileum, the final section of the small intestine.
So the complex travels all the way to the ileum.
All the way. There, it binds to its receptor on the apical membrane of the ileal enterocytes. The whole complex is then taken into the cell by endocytosis.
And inside the cell.
Inside, B12 is released from IF. It then binds to another protein, transcobalamin 2, CDI. It's this B12 TCI complex that exits the basolateral membrane into the portal blood, heading for the liver and the rest of the body.
Wow, that is intricate. So many potential failure points.
Exactly. The classic example is pernicious anemia. It's an autoimmune disease that destroys gastric parietal cells. No parietal cells means no acid and, crucially, no intrinsic factor.
Without IF, B12 can't be absorbed in the ileum.
Correct, leading to severe B12 deficiency, causing megaloblastic anemia and potentially irreversible neurological damage. Other causes could be surgical removal of the ileum, Crohn's disease affecting the ileum, or even bacterial overgrowth in the small intestine where bacteria consume the B12 before it can be absorbed.
A really complex pathway with significant clinical implications. Let's switch gears slightly to minerals. Calcium.
Calcium absorption happens via two main routes. First, there's an active transcellular pathway that occurs only in the duodenum. This is the pathway regulated by vitamin D.
How does vitamin D regulate it?
Vitamin D basically stimulates all three steps of this pathway: the entry of calcium through channels on the apical membrane, the synthesis of a calcium-binding protein called calbindin inside the cell, which buffers the calcium, and the active pumping of calcium out of the basolateral membrane by a Ca2+ pump and an ACA exchanger.
So vitamin D boosts active absorption in the duodenum. What's the other route?
The second route is passive, paracellular diffusion. This happens between the cells, driven by the concentration gradient, and it occurs throughout the small intestine, jejunum, and ileum, as well as duodenum.
And this passive route isn't regulated by vitamin D.
Not directly. And interestingly, although the active transport is tightly regulated, most of the total calcium absorption over the entire length of the intestine likely occurs via this passive pathway, simply due to the larger surface area involved.
Huh, okay. What about iron? That's another critical one.
Iron is vital, mainly for hemoglobin. Absorption is generally quite low, maybe only 10–20% of what you ingest, but it's very tightly regulated. This is because, unlike calcium, we don't have a regulated pathway to excrete excess iron. So controlling absorption is key.
How is it absorbed? I know there are different types, like heme and non-heme iron.
Right. Let's focus on non-heme iron, the main form in plant foods and supplements. It's usually ingested as ferric iron, F3+ Tor. First step, primarily in the duodenum, is reduction to ferrous iron, F2+ size, by an enzyme called DCEAD on the apical membrane.
Why reduce it?
Because the main transporter for uptake into the cell, DMT1, divalent metal transporter 1, specifically transports Fe2+ Psi, not Fe3+ Psi, and DMT1 is another H+ Ti-Sas co-transporter, more secondary active transport.
Okay, F2+ inside the cell via DMT1.
Inside, F2+ might bind to a protein called mobilferrin, though its exact role is debated. To get out of the cell across the basolateral membrane, F2+ uses another transporter called ferroportin, FP1.
So DMT1 for entry, ferroportin for exit.
Exactly. And just outside the cell, associated with ferroportin, is another enzyme called hefastin. It oxidizes the E2+ back to E3+, so it can bind to transfer, the main iron transport protein in the blood.
What about heme iron for meat?
Heme iron absorption is actually more efficient. Heme itself is taken into the enterocyte, possibly by a specific transporter. Inside the cell, an enzyme called heme oxygenase breaks open the heme ring, releasing F3+ of that. This iron is then reduced to F2+ and joins the same intracellular pool as the non-heme iron, exiting via ferroportin.
So problems with iron absorption lead to?
Iron deficiency anemia, the most common nutritional deficiency worldwide. But you can also have the opposite problem, hemochromatosis.
Too much iron.
Yes, it's a genetic disorder of excessive iron absorption. The body absorbs iron inappropriately even when stores are high. This is often linked to defects in the regulation by hepcidin.
Hepcidin, what's that?
Hepcidin is a hormone produced by the liver. Normally, when iron stores are high, hepcidin levels rise, and it acts to decrease iron absorption by causing the degradation of ferroportin, trapping iron inside enterocytes, which are then shed. In many forms of hemochromatosis, this hepcidin regulation is faulty.
And conversely, you mentioned anemia of inflammation.
Right. During inflammation, cytokines like IL-6 stimulate the liver to produce more hepcidin. This leads to decreased iron absorption and iron release from stores, contributing to the anemia often seen in chronic inflammatory diseases. It shows how iron metabolism is tied into the immune system too.
Really complex regulation. Okay, let's zoom out quickly. What about overall nutritional requirements? Do we need carbs and fats every day?
Well, not in the same way we need vitamins. There's no strict minimum daily requirement for carbs or total fat, assuming you get enough calories. However, we absolutely do need essential fatty acids.
Linoleate and linolenate, omega-6 and omega-3.
Exactly. We can't synthesize these, and they're precursors for vital signaling molecules like prostaglandins, thromboxanes, leukotrienes, so you can't have a completely fat-free diet long-term.
In protein, there is a requirement there, right?
Definitely. Adults generally need about 0.8 grams of protein per kilogram of body weight per day. Needs are higher for growing children, pregnant women, athletes, people recovering from surgery or illness.
And it's not just about quantity, but quality too, the essential amino acids.
Absolutely critical. There are 9 essential amino acids that our bodies cannot make, so they must come from the diet. If even one is missing, protein synthesis is limited.
And protein does more than just build muscle, right?
Oh, much more. tissue maintenance and repair, enzymes, hormones, neurotransmitters, and crucially, host defense. Think antibodies, immune cells, the integrity of your skin, and mucous membranes all rely heavily on adequate protein.
Right. And vitamins and minerals, generally they aren't energy, but essential cofactors.
Exactly. Integral to metabolism, immune function, nerve conduction, muscle contraction, blood clotting, you name it. Deficiencies, especially vitamin deficiencies, can cause pretty significant functional impairment relatively quickly. Mineral deficiencies often take more extreme dietary lack, but are equally serious when they occur.
What about taking too much? Megadoses.
It's a mixed bag. Small excesses of some nutrients, maybe like vitamin E or zinc, might offer some benefit in specific situations like stress, perhaps boosting immune response slightly, but truer excessive intake can definitely be harmful.
Like hypervitaminosis A from eating, say, polar bear liver.
That's the classic extreme example. But even high doses of supplements can be toxic. Too much vitamin D, too much iron, especially in kids, even too much calcium can interfere with the absorption of other minerals like iron and zinc.
And there's a difference between water-soluble and fat-soluble vitamins in terms of toxicity.
Generally, yes. Excess water-soluble vitamins, like C and most B vitamins, are usually just excreted in the urine, making them harder to overdose on, though not impossible with massive doses. Fat-soluble vitamins, A, D, E, K, are stored in body fat and the liver, so they can accumulate to toxic levels much more easily.
So, a lot to consider. We've covered a huge amount of ground.
We really have, from the initial breakdown of carbs, proteins, and fats to the intricate absorption of vitamins and minerals.
So, what does this all mean for you, the listener? We've journeyed through these incredibly detailed yet, remarkably efficient and adaptable mechanisms.
And understanding this intricate dance, all these enzymes, transporters, pathways, it isn't just for exams. It's absolutely crucial for diagnosing why someone might not be absorbing nutrients properly, for developing targeted treatments, even for making smarter dietary choices in your own life.
Yeah, this fundamental physiological knowledge really shapes your health, literally one nutrient at a time as it crosses your gut lining.
It's the foundation.
Absolutely. Remember, you're part of the Deep Dive family, and the more you unravel these complex systems piece by piece, the more confident you'll become. You are absolutely capable of mastering this material. Keep diving deep.